The purpose of this slide deck is to provide an example of a toy model for which we can practice explaining the distribution of instantaneous copy numbers of a molecule by regarding the synthesis of the individual molecules as stochastic events. The assumption that messenger RNA has a lifetime connects the probability distribution of transcription events occurring over time intervals to the distribution of copy numbers observed at individual times. This slide deck is divided into two videos. Our goal is to describe the statistics of copy numbers of messenger RNA in a simplified cell in which messenger RNA transcription and degradation are both stochastic. Messenger RNA copy numbers will turn out to be Poisson distributed, as we will demonstrate in the second video. To provide foundational intuition for this result, we will first explore a related but even further simplified model in which messenger RNA transcription is stochastic but degradation is deterministic. We are inside a cell. A copy of RNA polymerase repeatedly encounters a sequence of DNA, occasionally succeeding in producing messenger RNA but much more often than not, failing to initiate transcription. A failed attempt at transcription could be the consequence of geometric distortions in the copy of RNA polymerase or, for example, the consequence of incorrect orientation between the copy of RNA polymerase and the targeted sequence of DNA. Many unproductive collisions may take place between rare instances of transcription. The copy of RNA polymerase and the targeted gene forget, so to speak, their relative orientations as they circulate and bump into other molecules inside the cell before encountering each other again. The collisions we have animated are meant to be statistically independent. We concisely represent this cartoon using a wheel of fortune. The outcome of spinning the wheel once is a model for the outcome of a bunch of encounters between the copy of RNA polymerase and the gene of interest. If the needle points in the blue sector, then not a single transcription event has resulted from the bunch of molecular encounters represented by the spin. If the needle points instead in the yellow sector, a single transcription event has occurred. In principle, a bunch of molecular encounters could result in a bunch of transcription events. Given, however, the low probability of getting even a single transcription event, we neglect the possibility of these other outcomes. This is why the wheel lacks the additional sectors that would be necessary to indicate situations in which 2, 3, or more transcription events had resulted. Examples of times at which messenger RNA is transcribed can be simulated by spinning a collection of wheels arranged along a timeline. For example, one of the molecular encounters during the time interval represented by this wheel might have succeeded in initiating transcription, meaning that the wheel landed in the yellow sector. The resulting strand of messenger RNA then persists in the cell for some time before it is degraded. We label this time interval T-survive, and we simplify this initial discussion by assuming that all copies of messenger RNA survive for exactly the same duration. How many copies of messenger RNA are inside the cell? The answer depends on the time at which we count the copies of messenger RNA. For example, at this value of T count, we would count 2 copies of messenger RNA. The timeline on this slide also includes examples of times when the cell contains only 1 copy of messenger RNA and times when the cell contains no copy of messenger RNA. Please point to these times by pausing the video now. We want to describe the statistical distribution of messenger RNA copy numbers that we encounter when we sweep T-count across not only the times displayed, but also many other times beyond the left and right edges of the slide. To do so, we recognize that the number of copies of messenger RNA counted at the dashed line equals the number of messenger RNA molecules that were produced within an immediately preceding time interval of duration, T-survive. Messenger RNA molecules produced before the time interval indicated by the pink brace cannot contribute to the copy number of messenger RNA molecules measured at time T-count because these molecules degrade before they are counted. Messenger RNA molecules produced after the pink brace cannot be counted because they become present only after the count has already been completed. The number of messenger RNA molecules at time T count equals the number of transcription events that occur within the pink brace. In other words, the copy number at time t count equals the number of these wheels that land on yellow. Viewers who have recently reviewed the unit on the Poisson distribution should be familiar with this situation. Each wheel represents a bunch of molecular encounters. We can approximate an individual wheel by replacing it with two wheels that each represents half as many molecular encounters and thus has half the chance of producing a transcription event. If each wheel thus generated still represented many molecular encounters, subdivision could occur many times, yielding collections of wheels with ever more slender yellow sectors. Subdividing wheels mitigates an error depicted in this cartoon. The pink brace indicates a time interval whose boundaries slice through two wheels. In this case, both of the wheels are situated more outside rather than inside the pink brace, so we pretend that they do not contribute transcription events to the time interval of interest. In a sense, we are introducing rounding error. By subdividing time into ever shorter intervals, the errors introduced by these edge effects are reduced. But this is an aside. The point is that we can subdivide to get larger collections of wheels of fortune with, individually, smaller chances of landing on yellow. This is the limit of rare events. Thus, the number of transcription events occurring within the pink brace is Poisson distributed. Because the number of transcription events in the pink brace is equal to the number of copies of messenger RNA counted at time t count, the messenger RNA copy number is also Poisson distributed. When we say that the number of transcription events in the pink brace and the copy number at time t count are Poisson distributed, we are referring to the varied numbers of transcription events and molecules that we could record, for example, by sweeping t count across time in a single cell, or, for example, by keeping t count fixed to one time value but looking at a population of cells. Obviously, the one well-defined copy number of messenger RNA molecules at the one well-defined time t count in this one cartoon has only one value, and in this case it's 2 copies of messenger RNA. On this slide it has no other value and thus does not fill out a Poisson distribution. The assumption that messenger RNA has a lifetime connects the probability distribution of transcription events occurring over time intervals to the distribution of copy numbers observed at individual times. In this video, we connected repeated independent attempts at messenger RNA transcription to Poisson-distributed messenger RNA copy numbers. We assumed that all copies of messenger RNA survive for precisely the same duration. Now that we have established intuition using this simplified model, we can study a more realistic model in which both transcription and degradation are stochastic. In the next video, we will find that this more sophisticated model also leads to the conclusion that messenger RNA copy numbers are Poisson distributed.